Welcome! Today we'll be presenting about Ocrelizumab, a new drug for the treatment of multiple sclerosis.
Ocrelizumab is a monoclonal antibody drug used to treat multiple sclerosis. Being approved to treat relapsed-remitting MS and primary progressive MS, the drug targets B cells as they play an important role in the pathogenesis of MS by activating other immune cells and increasing inflammation in the brain and spinal cord. Ocrelizumab functions by binding on B cells and preventing them from crossing into the brain where it will attack the myelin sheath. The target for ocrelizumab is the protein CD20, which is found on all B lymphocytes. It was discovered in 1980. The protein is expressed in late pre-B lymphocytes, but not in pro-B lymphocytes, and as the cell matures, the protein is no longer expressed, being lost in differentiated plasmablasts and plasma cells. CD20 plays an important role in development and differentiation of B cells into plasma cells. CD20 is encoded by the MS4A1 gene, found on chromosome 11 in humans. It is a member of the MS4A family, being named for its 4 hydrophobic transmembrane domains, with 2 extracellular loops, one small and the other large, and also has an intracellular loop. The N-terminal and C-terminal of the CD20 protein are both in the cytosol. The protein exists as a homodimeric and homotetrameric oligomer on the cell surface of B cells and associates with other proteins to contribute to signal transduction. Ocrelizumab binds to a small section in the large extracellular loop of CD20, rendering the B cell non-functional. The precise function and regulation of CD20 is still unclear, as homozygous mutation of MS4A1 gene in humans is extremely rare. However, there is still some research which have been conducted on homozygous individuals. It was found that lacking CD20 did not disrupt the differentiation of pre-B cells in the bone marrow. However, this CD20 deficiency resulted in a reduced level of circulating memory B cells and decreased levels of IgG antibodies. Studies in murine CD20 deficient B cells suggest that CD20 is required for optimal T-independent humoral immunity and response to T-dependent antigens.
Now we move on to the next portion of this video, which is drug mechanisms. Ocrelizumab is a humanized anti-CD20 monoclonal antibody that depletes and inhibits the growth of CD20-positive B lymphoma cell lines. This is seen in Figure 1, where Ocrelizumab, the antibody, attaches to the CD20 molecule present on B cells. Ocrelizumab, like most existing anti-CD20 antibodies, are categorized as type 1 antibodies. Type 1 antibodies exhibit specific characteristics which affect their mechanism of action. I will touch upon this much later in the video. Although the exact etiology of B cell depletion is unknown, 3 different mechanisms of action have been suggested. The first one is complement-dependent cytotoxicity. The second one is antibody-dependent cellular cytotoxicity. And the third one is apoptosis. Compared with rituximab, which is known to be the first therapeutic B-cell depleting monoclonal antibody that showed efficacy in multiple sclerosis clinical trials, Ocrelizumab is associated with increased antibody-dependent cell-mediated cytotoxicity effects and reduced complement-dependent cytotoxic effects in vitro. The first suggested mechanism is complement-dependent cytotoxicity. Complement-dependent cytotoxicity is the mechanism by which antibody-coated target cells, which in this case would be the anti-CD20 monoclonal antibodies, recruit and activate components of the complement cascade. Complement-dependent cytotoxicity is initiated when C1q, the initiating component of the classical complement pathway, is fixed to the Fc portion of target-bound antibodies. This activates multiple steps within the cascade, which ultimately results in the formation of the membrane attack complex on the cell surface. This is shown very clearly in Figure 2, where the membrane attack complex is labeled as MAC and is a triangle. The formation of MAC subsequently results in cell lysis by forming pores in the cell. Coming back to the function of type 1 antibodies, they are characterized by their ability to induce a translocation of CD20 into large lipid microdomains or lipid rafts within the plasma membrane upon binding. This clustering process enhances the recruitment and activation of complement, and hence type 1 antibodies exert potent complement-dependent cytotoxicity. The second suggested mechanism is antibody-dependent cellular cytotoxicity. This is an immune mechanism through which Fc receptor-bearing effector cells recognize and kill the anti-CD20 monoclonal antibodies coated B cell expressing CD20. As can be seen in Figure 3, effector cells may include macrophages and natural killer cells that act together to cause this cell destruction. The last suggested mechanism is apoptosis, which may result from cross-linking membrane CD20 on the target cell surface. Apoptosis is a form of programmed cell death. It has been proposed that increases in intracellular calcium 2+ levels, which occur in response to anti-CD20 cross-linking, may be involved in the apoptotic process, but the precise mechanism is still unclear. The diagram in front of you displays the actions of cross-linking. Another characteristic of type 1 antibodies that allow these antibodies to exhibit their effects is that twice the amount of antibodies can bind to B cells. This is most likely due to different binding geometries. This therefore causes an increased chance of cross-linking.
We will now be looking at the pharmacokinetic properties of Ocrelizumab. Ocrelizumab is given via an intravenous injection. It follows a two-compartment pharmacokinetic model, which means that Ocrelizumab moves between the central and peripheral compartments of the body. Due to this, peripheral and central clearance rates may vary. Ocrelizumab is initially given via two 300 mg doses. The second dose is only administered 14 days later. After that, Ocrelizumab is given every 24 weeks with a single dose of 600 mg. The infusion time can take over 3 hours, and antihistamine medication may be given before the infusion. Ocrelizumab has potential interactions with vaccines due to its immunosuppressant qualities. Vaccine jabs are not recommended during Ocrelizumab therapy and should be given only after B-cell repletion occurs. Further studies are required to understand the full interactions of Ocrelizumab and vaccines. It also interacts with both immunosuppressant drugs and other MS drugs such as teriflunomide. In both cases, the interactions may compromise a patient's immune system. Since Ocrelizumab is injected intravenously, it has 100% bioavailability. It also has a high central volume distribution of 2.78 liters. This indicates that Ocrelizumab has a strong tissue binding and is lipid soluble, allowing it to pass cell and blood barriers easily. Ocrelizumab binds to B cells in our blood and can also be found in our lymphatic liquid. Since Ocrelizumab is injected intravenously, it has 100% bioavailability. It also has a high central volume distribution of 2.78 liters. This indicates that Ocrelizumab has a strong tissue binding and is lipid soluble, allowing it to pass cell and blood barriers easily. Ocrelizumab binds to B cells in our blood and can also be found in our lymphatic liquid. Antibodies are far too large to be filtered via kidneys, so the renal system doesn't play a large part in either excretion or metabolism of Ocrelizumab. Instead, it is catabolized into peptides and amino acids, just like the rest of our antibodies. This degradation can happen in our liver via proteolysis, where proteases hydrolyze the peptide bonds of the antibodies and break them down into smaller parts. The antibodies may also be taken up into cells via non-specific endocytosis. After the antibodies are taken in, they are broken down by lysosomes in our cells. Ocrelizumab isn't excreted via urine nor feces, since it is injected intravenously. It does, however, have a clearance rate of 0.17 liters a day and a terminal elimination half-life of 26 days. Terminal elimination looks at the time required for the plasma concentration of a drug to fall by 50%, which is a better way of looking at ocrelizumab's half-life since it is not excreted. Ocrelizumab effects can last for up to 3 years after the last injection, and is potentially excreted into breast milk. However, more research into this needs to be done as it has implications for the health of a growing baby.
The adverse side effects of ocrelizumab in relapsing MS with an incidence of more than 5% and higher compared to the other MS drug interferon beta-1a include herpes virus-associated infections, depression, lower and/or upper respiratory infections, and pain in the extremities, to name a few. The adverse reaction in primary progressive MS include coughing, skin reaction, and lower or upper respiratory infection are one of the few side effects associated with the drug. There are contraindications for ocrelizumab, as patients with hepatitis B cannot receive the drug as there is a chance that it could reactivate the latent virus, as it is an immunosuppressive drug. There are some side effects that occur during injection or up to 24 hours later. Patients will start experiencing faster heartbeats, nausea, dizziness, throat pain, feelings of shortness of breath, flushing, and nerve pain such as tingling or pins and needles. Some of these symptoms, however, are common with any type of injection. It is not known whether the drug can cause adverse fetal harm or reduce the reproductive capacity in humans. However, in animal studies, doses equivalent to human or more have shown to increase perinatal mortality, depletion of B-cell population, and bone marrow and testicular toxicity were observed in the offsprings. Ocrelizumab can be used during pregnancy only if the benefit outweighs the risk, as the drug is a humanized monoclonal antibody of an immunoglobulin G1 subtype that does not cross the placental barrier.
Thank you for listening to our presentation. Here are all the references cited in Harvard.